So it's a great honor and pleasure to be here. And yeah, there were several postponements. Initially it was May 2020, and then it was May 21, and now it's November 21, but I'm here, so that's great. Okay, so what I'll do today, sounds a little loud to me, but he can adjust it. What I'll do today, I'll first give you a sort of an introduction to the system and an overview, because it's sort of a complicated system to some extent, and then towards the end, I'll show you also some really exciting unpublished data that actually were very unexpected when we did the experiments, and they led us on new avenues that are likely to keep us busy for a few more years. So just using this sort of, actually there was a cover of Nature Cell Biology, but what you can see that, to sort of start the talk, that what we're going to talk about is the polarity in the plane of a tissue. So it's either in the plane of an epithelium, if it's an epithelial tissue, but it could also be non-epithelial cells that develop in a plane. And you can see that, for example, this patch here that's labeled in green is secreting a Wnt molecule, and it makes the cells that are surrounding it orient towards that green patch. So that's sort of the theme that we will be trying to understand how cells orient in the context of an epithelial tissue or a tissue in general. And so the bigger chunk of the talk will be on Wnt-frizzled Pcp signaling, and in the end I will also speak about the canonical Wnt Beta-Catenin signaling. And the reason why it's really good to study this in Drosophila, because of course we have the genetics, but that we had, even 20 years ago. Now with CRISPR you can do a lot of genetic stuff in a sort of sophisticated way in other organisms as well. But it's also the ease of observation, that we can really see stuff very easily. And this is just 3 examples of tissues that we usually study. This is a section through the facet eye, the Drosophila eye. And you can see these units, the facets, the ommatidia, are very precisely arranged in the equatorial polar axis, in the anterior-posterior axis. This is a blow-up of a part of the wing, where each cell has an actin hair that's pointing proximal to distally. And this is a sort of a low microscopic view of the thorax, anterior to posterior. You can see there's all these sensory bristles, which are multicellular organs. They are aligned in the anterior-posterior axis. And again, just to put everybody on the same plane. So the big chunk of my talk will be on frizzled Pcp signaling, and to sort of clarify, downstream of one of the effectors, disheveled, which is still a very enigmatic protein, and Vita knows more about disheveled than probably most people in the world, besides maybe me and Sergei Sokol, but it's a crazy protein, so it'll keep all of us busy for a few more years. It's a different pathway. This is the canonical pathway. In the most simplest view, you have a destruction complex that affects beta-catenin. It goes in the nucleus to activate transcription. Here it's more complicated. You have different ways of activating downstream effectors, and I'll come back to that later. And again, then what we're studying is the plane. So how cells get oriented and polarized within the plane of the epithelium, so they know anterior, posterior, left, right, dorsal, ventral, etc. And what you see here, this is the same picture as before, but what I highlighted here is again the axis of polarity. So we have, in each of these tissues, we have an axis of polarity, and this is depending on the tissue. It's proximal distal, anterior posterior, or more complicated, for example, in the eye, it's from the center to the dorsal and the ventral pole. And we study this in flies because, again, this is the best system to study it, but these are several mammalian and vertebrate systems where Pcp, the same molecular cassette that I'll introduce, is also regulating processes. So it was initially shown, which was actually, again, in non-epithelial tissues, in gastrulation invertebrates, convergent extension during gastrulation, that it has the same players are critical there. And this was shown in xenopus initially and then confirmed in zebrafish. But all these other cells are kind of epithelial of origin in many cases. So it's, for example, the inner ear polarization. Skin, our skin is polarized. Left-right definition in the body. Female reproductive tract needs to be polarized. Respiratory system, the cilia in the respiratory system need to be properly placed. Axonal pathfinding uses the Pcp system. And cells that, cancer cells during metastasis. And the whole sort of set of ciliopathies that are cilia-associated diseases rely on Pcp signaling because Pcp positions the primary cilium. So if you misposition the cilium, you end up with a form of several of these ciliopathies. So what I'll do first, I'll sort of introduce the players, and then I'll show you some of the data. So the core of the Pcp factors, again, is based on the Wnt-frizzled interaction, but it's more complicated here because you don't just activate the pathway, you have to activate the pathway within the cell asymmetrically. So you have a complex that is anchored by frizzled disheveled, and it also induces colocalization of Diego, which is the Drosophila homologue of inversin, which comes back to the left-right asymmetry, as I already mentioned. And this whole thing is sort of also anchored in the membrane by a very atypical cadherin. It's a 7-pass transmembrane cadherin that mediates homophilic cell adhesion. And on the other complex, which induces sort of the specific localization of frizzled disheveled, is anchored by Wang, a 4-pass transmembrane protein with prickle and associated factors. And so these two complexes don't like each other within the cell, so they try to go to the opposite end. And the interaction between frizzled and Wang between cells also stabilized and kind of strengthened by the interaction between the flamingo proteins that associate with frizzled or Wang is then also propagating the signal from cell to cell and stabilizing these complexes between cells. So you have a stable interaction between cells and a antagonistic interaction within the cell. And so when you look at the whole tissue, this is sort of a slightly lower magnification view. So it's propagated from cell to cell, and the initial bias or cue for polarization comes from Wnt expression or Wnt gradient that basically pushes frizzled or stabilizes frizzled more on one side of the cell, and that then triggers the localization. But very often when you localize some of these proteins, you will affect neighboring cells, not just the cell that is mutant, because through this cell-to-cell interaction propagation, you basically need cells to talk to each other. So if one cell makes a mistake, the neighboring cell can be influenced by it. And again, so I will now focus mostly, this is just for clarification, what we are going to discuss mostly is how frizzled and disheveled get localized. But frizzled disheveled in PCP activates several pathways. Most prominently, cytoskeletal arrangements are regulated asymmetrically within the cell, but it can also affect nuclear signaling through the J&K MAP kinase cascade affecting Jun and Phos. So there's sort of a variety of branches of signaling downstream of frizzled disheveled in PCP as compared to canonical signaling, which is a simple it's not simple, but it's more straightforward. It's basically activation of a certain component of the pathway that goes to the nucleus, and that's it. So here it's more diverse depending on the tissue. And before I go into the more detail, I'll just show examples why we really can say that this is so similar between Drosophila and vertebrates. So this is the flywing, wild-type, frizzled mutant. This is a lucky case in the mouse where a single mouse frizzled knockout, this is mouse frizzled 6 from Jeremy Nason's lab, causes skin phenotypes. This is the neck, the dorsal neck, that are very similar to the wing phenotypes in the fly. Again, this is the wild-type for comparison. So in most cases in the mammalian system, you need to make double or triple mutants to see something because there are many frizzles, there are three disheveleds, etc. And this is now an example. Again, the eye is a good example for a neural epithelial structure, the retina, that's polarized, so this will be wild type. And then the original frizzled one and disheveled one allele, the photoreceptors still form, they are all there, but they are misaligned with respect to the axes. And this is quite similar to our ear, so the mammalian inner ear, this is a mouse ear here, where we have wild type, where the sensory cells are very nicely aligned from the inner to the outer ridge in the cochlea, both in antibody staining or in the scanning EM. The loop tail is a allele of Wangel-like 2, sorry. And again, you can see that these cells look normal. They have a kinocilium, they have the stereocilia, but they just misalign, so they point sort of randomly within this axis. So it's similar, again, from Drosophila to mammals, and this is just examples that are more extreme. So this is now zygotic mutations in some of the core PCP genes, such as Wangel. Seltzer is the flamingo cadherin. Single mutants, double mutants, and here they affect the neural tube closure, which is sort of the second phase of convergent extension, where the tube closes after gastrulation has been completed. So again, just to highlight that it's acting also earlier in development before we can sort of look at these adult structures. So many, if not all, of the real sort of severe mutations in the PCP genes are early embryonic lethal. Okay, so we had that already. These are all the other examples. So I'll go a little bit more through this and then show you sort of inside how we look at this. I just like to highlight one thing here, that within this complex, the frizzled disheveled site will regulate positively actin polymerization by recruiting Rho GDPase family members acting on Rho kinase and other actin regulators, versus the Wang prickle site recruits a few proteins culminating in the recruitment of a gene called multiple wing hair, which is a antiformin. So and from its get-go, it antagonizes actin polymerization. So not only don't these complexes like each other, they separate each other, but they also have sort of opposite effects on the cytoskeleton. So there's, again, I said that already, I just repeat it again. So there are intracellular antagonistic interactions between these complexes, and there are positive feedback loops across cells, because once you have this as a stable complex, it'll be maintain itself and also propagate from cell to cell. And just as an example, why we can very precisely say which protein is localized in which side of the cell, this is endogenously tagged GFP tagged Diego, which would be the inversin, which depends on frizzled and disheveled for localization. So if you start in the late larval instar, where this is when the epithelium is starting to get patterned, you have basically uniform localization of the PCP genes along the adherence junction belt. And a day later, 24 hours later, they split. And so you have the Van Gogh strabismus prickle complex on one side, frizzled disheveled Diego. And in vivo, you can nicely visualize Diego is here found only on one side of the cell, which is the distal side and not the proximal. If you do the same experiment, for example, with prickle, it'll be on the opposite side. And if you do it in a mutant background, it'll be mislocalized because the complexes don't properly form. Because of these interactions, both the antagonistic and the positive feedback loops, this is a self-organizing system. So once you give it a clue which way they should orient, they will do that fairly readily in a sort of sequential way, starting with subtle polarization into very strongly polarized tissues. So it's important to keep that in mind, because again, when you make a mistake, the mistake is usually propagated because it'll kind of misorganizes the tissue in general. And this is just, again, an example to show that each of these factors depends on the presence of all of them. So if you remove any of these 6, the system falls apart because you need the interactions between all of them. The transmembrane proteins are a little bit more important because they clearly are the anchors of the complexes, but also the cytoplasmic proteins, if they are mutated, disheveled Diego or prickle, the complexes start falling apart. So in this context, this is a frizzled knockdown. This is the control tissue, stained for flamingo, and so you can see this as a fairly regular zigzag pattern. Also, the cells become hexagonal, nicely regular, and you can see the polarity arrows point from proximal to distal. And in the half of the wing here where we knock down frizzled, flamingo becomes mislocalized, so it's all around the cell. The cells have different shape, they're not hexagonal anymore, and the polarity vectors are randomly distributed, but they're also much shorter because there's just less polarity. Okay, so we discussed that. And so what I'll show you now, just to kind of, again, bring you back to the wind story. In Drosophila, it was difficult to visualize wind requirement for this PCP orientation because unlike for most genes in Drosophila, here we are looking at 2 redundant genes that are normally not redundant. So wingless obviously is not redundant, but for PCP it is with Wnt4. And it serves as an organizer, and I'll show you some data along those lines. And this is quite similar to Wnt5 and Wnt11 in the vertebrate context. Several labs have shown in the mouse and also in zebrafish and in Xenopus that if you position Wnt5 or Wnt11, you will start polarizing cells. So there is a requirement for the winds. And the best way to show it was looking at the fly wing, but we see the same in the fly eye. The wing initially is radially polarized. That's also one of the reasons why it was missed, because what we usually look at is this late-stage proximal distal polarity, but when it's established, it's radial, and there is wind expression along the margin, and it's wingless and Wnt4. And so when Suzanne Eaton established that actually polarity is already present at this stage, obviously it opened many people's eyes, and we started looking at it. And the Wnt genes, in this particular case, wingless itself and dWnt4, are nicely expressed along this margin. And so if you make a double mutant between Wnt4 a null allele, and then a wingless temperature-sensitive allele, because you have to keep wingless present so that you allow the tissue to develop. If you remove wingless early, there's no wing, as the name says, and so you can't even look at it. So you start seeing PCP defects, and if you look early, which is again at this stage when normally people do not look, Suzanne Eaton showed us that's the time to look. So in wild type, all the cells polarize towards the margin, so you can see these yellow polarity vectors pointing towards the margin here in this sector. And in a double mutant, you lose polarity, it becomes random with short polarity factors, polarity orientation and polarity strength. What's important, and it's sort of anecdotal, but it is important, if you go close to the wing margin where the winds are expressed, there's no polarity yet detected at this stage, because the cells that are sitting here, they're getting too much wind, so they are confused. The cells that are here, they see wind coming from here, so they have a sort of sense of orientation. And the cells that are near the margin, initially they get so much wind that they are kind of flooded by it, and so they don't know where the margin is. And so polarity in these cells gets only established later through these progressive interactions between these cells. And the same applies when you're really far away from the gradient to see that. And so the second aspect I'd like to emphasize here is that the winds are instructive for polarity. So this is again, in this hinge region, we're looking now on the dorsal anterior quadrant here. Normally cells are oriented towards the margin, and now here we misexpress wind 4. And if you look now at the cells that are just flanking this region, both inside and outside, the polarity is very tightly aligned, perpendicular at a 90-degree angle to the border of the clone. Because these cells, they sense the difference between wind-expressing and non-expressing cell just along this margin. So if you just look at the yellow dots and the blue dots, you get this very precise arrangement. Inside the clone, where you're inside the wind-expressing clone, it's random again. So you get the whole angle, and this will be wild type here pointing towards the margin. So it's both necessary and instructive. And this is again just that example. When we look at it at a later stage, when you start seeing the actin hair, you can see that going from proximal to distal, the proximal cells are fine because they point towards the tissue, but the distal cells, they would normally point that way, again, towards me, but because they see wind 4 coming from this side, they turn around and point into the clone. So this is just to, again, highlight this relationship with the expression of winds and the PCP axis. So the winds are necessary and sufficient to orient things. They're instructive. And in Drosophila, it's one of the few examples where you actually need to deal with redundancy. And because wingless is such an important gene, it's actually complicated genetics to really do that. And we can assume, based on, again, work from other labs, that Wind5, Wind11, and some of the other non-canonical winds do similar stuff, orienting cells in the limb or actually during regeneration as well. I was just in Vienna yesterday, and one of the people that works on regeneration, the axolotl, and she has nice Wind5 expression in the blastema, and regeneration orients itself in that axis as well. So that was quite nice. So just to summarize quickly, so wind expression serves as the PCP organizer. Core PCP protein levels need to be tightly regulated. So both when you have too much or too little of any of these 6 factors, the system falls apart. There's a certain range where things get regulated, but if you flood the system with too much of any of these factors, or you remove one, it goes off the rail. And this is, again, just to kind of summarize this here. The important aspect here that depending on the tissue, the axis of polarity is different. In the eye, we have winds expressed on the poles, so it's equatorial polar. In the appendages, like the wing or the leg, it's proximal to distal, again, because of the expression of the organizers. Okay, so for now, so far I've been only talking about these core factors. So now we're kind of interested in looking at what are these factors doing, how are they regulated to interact with each other. And you can imagine there's a whole range of regulatory proteins such as kinases, E3 ubiquitin ligases, and other proteins. So we were looking at a set of kinases, and here on the side of frizzled disheveled, we have CK1s, we have APKC, ABL, tyrosine kinase, NEC2, that phosphorylate either frizzled or disheveled and help stabilize or destabilize the complex. Similarly, on the Wang prickle side, we have another CK1 isoform, we have NEMO, other PKCs, And I'll tell you briefly about one of the kinases here that we haven't published yet that is involved here, that's SARC. And they, again, stabilize or destabilize these complexes depending if they're positive or negative regulators. So we did some work on frizzled and disheveled, but Wang is actually the most specific PCP gene in the genome. It seems to be acting really tightly in the PCP context. None of the others is as specific, so it seemed worthwhile to look more closely at Wang. There's also only 2 Wang-like genes in the mammalian genome, Wang1 and Wang2, so there's less redundancy in the mammalian genome, so it was kind of easier to focus on that. So we did a mass spec analysis on phosphorylation of Wang, and this was done also in parallel by Daniel Davenport at Princeton with mouse Wang2. And so there's a set of serines renins in the cytoplasmic N-terminal tail that's phosphorylated and important, and this is published, so I'm not going to go too much detail. This is phosphorylated by CK1 kinases. But what is interesting in the cytoplasmic C-tail, the C-terminal tail that sticks into the cell, we find 4 phosphorylated and highly conserved tyrosines. And again, one is published, so I'm just going to show you the data. So this tyrosine here is highly conserved, and it's required, its phosphorylation is required for membrane trafficking. So if you're mutant for this tyrosine, the Wangel protein is made, but then it gets stuck in the ER. It never makes it to the membrane. So the whole system also falls apart because if you don't have Wangel at the membrane, it doesn't work. This one is very interesting by its location. So if you look at the binding domains or binding sites for Diego, prickle, disheveled that have been finally mapped by Ashley Humphreys, a postdoc in the lab, prickle and disheveled map to this region flanking this tyrosine, and they even overlap in 2 amino acids exactly over this tyrosine. So we reasoned, and this tyrosine is 100% conserved in any Wangel protein you find in any sequenced genome. So it seemed reasonable to sort of look, does it affect prickle disheveled binding? And you can guess it does, otherwise I wouldn't tell you. So what's really interesting here is we mutate the wild type, the tyrosine, into phenylalanine. Prickle doesn't bind anymore. If we add charge, so charged amino acids instead of the tyrosine, prickle does bind, and depending on how much charge you put, it binds better or worse. Disheveled is the inverse. If we now make tyrosine into phenylalanine, disheveled will bind nicely. It doesn't like alanine, so it likes the aromatic ring of the residue. And again, if you add charge with the charged amino acids, it doesn't bind. So the simple conclusion here is that disheveled prefers aromatic ring, phenylalanine, to bind over charge, and prickle prefers charge, which basically means whenever this is phosphorylated, prickle will be preferentially binding, and this is reflecting the stable formation of the complexes. And when it's not phosphorylated, disheveled will be binding, which will be the unstable formation of complexes before it's resolved. So obviously, what's the kinase? And so looking at the sequences, the kinase that stood out, it looks most likely like a SARC site. And SARC does phosphorylate there in vitro, but that doesn't mean much because SARC will phosphorylate many sites in vitro. But if we start playing with the SARC kinase levels, this is now, SARC is difficult to work with, so I'll explain in a second. But we can, for example, express or knock down the C-terminal SARC kinase regulator, CSK, which phosphorylates SARC and inactivates it or activates it, depending what you do. By knocking down CSK, you have too much SARC activity, and then we start seeing PCP defects both in the eye and in the wing. So that suggests that SARCs are critical. We cannot make, at this point yet, this will require some work to make SARC loss of function because the SARC42 is cell lethal, so we lose the cells before we can look at. And SARC64 is sort of redundant with 42 in some context and has no phenotype. But when we overexpress SARC, we get phenotypes that resemble strong PCP defects, including some cell loss. And these strong PCP defects are suppressed by removing a copy of the Wang gene, suggesting that Wang is certainly involved in that context. So this is still sort of works in progress, but the data suggests that phosphorylation of SARC-like kinases or SARC itself at this tyrosine makes a distinction between prickle or disheveled binding. And so when the complexes mature, which would be at a stage when you have prickle bound to Wang, it should be SARC phosphorylated or phosphorylated by a SARC-like kinase. So this suggests again that there's more maybe to be found at the level of these kinases. And so, how am I doing in time? Okay. So for the next part, I'll try to focus on sort of the conundrum a lot of people are facing working on PCP, that depending on the tissue you look at, you end up looking at different things biologically. So PCP can regulate cell fate by the nuclear signaling to Jun and Phos, which is important, for example, in the fly eye, where you specify different photoreceptors. Most importantly, it regulates cytoskeletal arrangements and orientation, which is sort of the bona fide phenotype that you would expect. But it also affects cell adhesion and thus cell motility. It affects the orientation of mitotic spindle, so this is important both during organogenesis but also during regeneration. And it affects ciliary positioning. So if you look at the primary cilium in fish and in mice, it usually ends up, or it does end up on the side of frizzled disheveled, which again is sort of maybe a cytoskeletal readout, if you want to call it that. So Ursula Weber designed a screen, and together with some postdocs and students, they were basically thinking, okay, if we play with Prickle and Diego by changing their levels a little bit, not too much, just a little bit, we might be able to find modifiers that would fall into category either regulating these factors, so being modifiers at this level, or possibly being effectors in some of these tissue-specific contexts. So this is just a brief overview of the screen. So we remove one copy of Prickle, so we have half the dose of Prickle, which is normally sufficient, and we overexpress Diego a little bit, not too much, and then you start seeing phenotypes that are both the PCP in the eye and in the wing is disturbed, and then we can screen the genome by using a combination of deficiencies, then RNAi, shRNA, etc., to narrow it down to individual genes. And so from that screen, we found kinases, ubiquitin ligases, that all make sense. One of the kinases that came from this was, for example, NEC2, that is key in phosphorylating disheveled, as Vita has very nicely shown biochemically. But we also found cilia-associated proteins, or ciliopathy-linked proteins. And this was very confusing at first, or still is to some extent, because the tissue we screened in do not have cilia. So the epithelial cells, both for the eye and the wing, and generally the Drosophila imaginal disc epithelia, they lost the primary epithelium because things develop fast in the fly, so they kind of got rid of that. But we still are able to find cilia-associated genes in this screen, so suggesting that there might be non-ciliary function of these ciliary proteins in these epithelia. So that triggered two sets of experiments we did. One set was doing a systematic screen, looking at the whole ciliome, which is 120 plus genes in the fly, and basically just asking, does any of these cilia-associated genes have phenotype in the fly, wing, or the eye that can be linked to some function like a signaling pathway, and I'll come back to that in a second. And the other one was also to see, maybe there's a link between this actin hair formation, that is sort of the readout that we're looking at here, that forms downstream of frizzled disheveled, and ciliary positioning. So Chema Carvajal-Gonzalez, when he was a postdoc in the lab, basically asked the question, is there a relationship between the actin hairs and cilia? And the first indication came where he started looking at centrioles, which are basically the non-ciliary equivalent to basal bodies at the base of cilia. And what he could see, this is sort of a low resolution thing, but what he could see that at the base of these hairs, we very often found centrioles marked here by astrales. Here in a more sort of cleaner cut staining, SAS4 is one of these scaffold proteins that is required to make and maintain a centriole, labeling both centrioles in each cell, and they sit at the base of the actin hair. And if you look at it, how this evolves during PCP establishment, initially when there's little PCP, so PCP is already set up, but the actin is still non-polarized, the centriole sits in the middle. And as the actin gets more and more polarized, so here you start initiating the actin base hair, here it's formed. We enrich the centrioles in the distal quadrant, which is at the base of the actin. So centriole positioning follows maturation of PCP establishment, which is kind of consistent with the idea that mature PCP will position the primary cilium. And this is just a few examples of what happens when you mess up PCP. So here is again the wild type control, this is just a marker for the junctions with DLG. These are SAS4 and actin, and again, it's highly enriched in the distal quadrant at the base of the hair. If this is now a flamingo mutant, it goes back to the center. The hairs also don't get properly polarized, so the cells still make hairs. Sometimes they make two hairs, sometimes they make sort of a broad hair without much focus. Centrioles are mislocalized. Frizzled mutant, centrioles are mislocalized, and this is frizzled overexpression. Centrioles are mislocalized, and you get PCP defect. So in both gain and loss of function PCP, consistent with the idea that you need the right level, centrioles get mislocalized. And the other question we then asked, this is also published, so I go through it a bit quickly. If you have cells that do not have not have centrioles, so we can make cells that are centriole-less by manipulating them genetically. PCP is still normal, which clearly puts the centrioles downstream of PCP and not the other way around. And what's quite intriguing and sort of cute here, that when you make cells that have multicentriolar with 5 or 7 centrioles, by overexpressing astrolase, which serves as one of these scaffolds. For example, here you have 5 centrioles, and they again enrich very tightly in the quadrant just under the hair. And this is just comparison to wild type, where we have the proper number of centrioles. This 5 or 7 centrioles, it's equally good. So all these data taken together suggests very strongly that there is a relationship, so that the actin hairs, because they kind of originate where the centriole is positioned, and that they also help position the centriole, is similar to positioning cilia. And so one of the key conserved PCP effects or readouts would be the positioning of centrioles, centrosomes, or basal bodies. And again, centrosomes would be for the spindle orientation, basal body for the cilia. And this is an example from a paper from Brian Ciruna's lab, where they were looking in the AP axis in zebrafish. You can see that the cilia are very tightly localized to the posterior side here, which is the frizzled disheveled containing side. So this is all consistent. Okay, so that's one of the indications that there is a relationship between a strong relationship between PCP and cilia and centrioles. And the second one, we basically were curious, so what do these ciliary proteins do in these non-ciliated cells? So to that extent, we did, again, I said we screened the whole ciliome. Ursula focused on the PCP readouts, and it looked like there's several proteins that fit into the ciliary transition zone group, anchored by MKS1 and possibly CEP290, that somehow affect PCP and possibly cell adhesion. This needs to be analyzed in more detail. But what was very surprising and unexpected at all, we pulled out IFTA, which is one of the ciliary transport modules that is required for intraflagellar transport, as it was named in Chlamydomonas, where it was discovered. But it's basically the adapter complex that binds to cargo and moves it up and down the cilium. Again, this was in non-ciliated cells, so what does IFT do outside the cilium, which was completely unexpected and novel. So this is, we published the screen, and I'll summarize it quickly. Initially, we could show that out of these 120 plus genes that are cilia associated, only a very small number have, in our assays, where we looked at all signaling pathways, Wnt, DPP, BMP, EGFR, RTK, Notch, JAK-STAT, etc. Only the IFT complex affected Wnt signaling, and 2 other genes affected Notch signaling, which we haven't followed up yet on. So we're focusing now on trying to figure out what IFT complex is doing in the cytoplasm at the level of canonical Wnt signaling. Oops, that's not good. That's fine. So just again, to put everybody on the same page, canonical Wnt signaling is different because they're simply over-exaggerated. You activate a pathway that has a repressive function. So there's a destruction complex anchored around axin, which is a scaffold that brings kinases in the vicinity of beta-catenin. Beta-catenin gets phosphorylated, degraded, and the pathway is repressed. The TCF transcription factor associates with co-repressors. If you add Wnt, you make a co-receptor-receptor complex, frizzled LRP56, which will recruit axin and dishevel to the membrane, form this as a signalosome. Beta-catenin is released from this complex. It's not phosphorylated. It'll accumulate, and it goes into the nucleus. But as I said, it goes into the nucleus, but how does it go into the nucleus? It does not have a proper NLS. Beta-catenin is one of the few proteins, or one of the set of proteins that goes into the nucleus without a canonical nuclear localization signal. So it's always a sort of a black box in that context. So let's see what I can show you that kind of hopefully will convince you that we have found a very intriguing new mechanism. So we went back to the wing using the canonical pathway where wingless is expressed at the margin and induces different sulfates in a gradient-dependent manner across the wing blade. And so again here, this bright red is wingless, and then we have these cells that require high levels of wingless, which will give rise to the sensory organs along the margin, which are marked by senseless. Then red is an intermediate target, distalless, and then other targets as well. So in that context, we can, for example, just to show you an example from the screen, we can knock down any of these ciliary proteins specifically in one compartment, here in the posterior compartment marked by blue. And so this would be the wild-type control, where you see these 2 rows of senseless positive cells. We knock down IFTA in the posterior compartment, and you lose the Wnt target gene expression. You lose wing senseless completely, and you reduce distalless, and this is quantified here as well. What's quite nice is a positive control or negative control. Not every IFT does that. IFT 144, where we know we can show IFT-related phenotypes in sensory cells, has no effect. So this sort of also is a nice control, because not every of these guys is doing the same, but 4 of them do the same. And so when you look at the IFTA complex, it's normally composed of 5 core proteins highlighted here. And of those 5, 4 show identical phenotypes when we knock them down in the context of Wnt signaling. And they seem to be basically required for regular canonical Wnt signaling. Just again, a reminder, IFTA is required for intraflagellar transport in Chlamydomonas, or for transport along the axonemes, which are the microtubules inside the cilium. So the IFTA complex is required for retrograde from the tip back to the base. IFTB goes from the base to the tip. And they basically interact with motor proteins binding to microtubules that move along microtubules, and then they bind cargo proteins that need to be transported in and out of the cilium. So having this in hand, then obviously the question was also, are there motors like kinesin, etc. involved? And Lin Wang, who joined the lab after she completed her PhD in Guang Choi's lab, she actually had already indication that kinesin is involved. So it was perfect for my lab to have her join, because she kind of knew the system. And so what she did here, she looked in more detail. She made clones of either an IFTA component, this is IFT 140, and you lose, again, senseless in the mutant cells that are marked here by absence of green. The kinesin shows the same phenotype. So kinesin 2, this is the Drosophila kinesin 2 homolog, has the same phenotype. If you make a double mutant clone, IFTA and kinesin, it looks the same again. And distalless is reduced, so it behaves like the experiments we did in the screen, where you basically do a knockdown of these structures. So we then asked the question, genetically, can we place it in the pathway? Can we put it upstream, downstream of certain well-known components through genetic epistasis? So if we, for example, take axin, which is one of these scaffold proteins in the destruction complex, and you make an axin mutant clone marked here by the absence of red, you see increase of armadillo, Drosophila Beta-Catenin, and in that context, you start seeing almost every cell in a certain range from the source will express senseless. So it basically, you get indiscriminate full activation of the pathway. If we now do a double mutant clone between axin and kinesin, shown here, or the equivalent with axin and IFT 140, not shown here, you get a very interesting phenotype. So again, anything that's minus or negative for red is axin minus. So inside the axin mutant cells, you get high levels of beta-catenin, because the destruction complex doesn't assemble, so you get lots of beta-catenin. And here now, part of this, the sister clone here, is double mutant for kinesin, and the green mutant, red and green mutant is double mutant, so it has a lot of beta-catenin, but it doesn't have kinesin, and senseless is not activated. In the neighboring clone here, it is activated, so it shows that the experiment works nicely. So this suggests very strongly that beta-catenin requires IFT, kinesin, and associated proteins downstream of the destruction complex, and so it could be involved in nuclear translocation of beta-catenin armadillo, or some other aspect that's after it gets released from beta-catenin. So the genetics clearly argue that kinesin to IFTA complex acts downstream of the destruction complex. So the question is, what is it doing downstream of the complex? And so we looked in more detail at other proteins that are associated with kinesin. Between IFTA and kinesin, you often find a protein called CAP3 for kinesin-associated protein 3 as another adapter. And CAP3 shows the same phenotypes as kinesin or IFTA, so it behaves very much the same in this context. And so what Linden started looking, both molecularly, biochemically, but also in co-localization, do these things localize together in response to Wnt signaling or in general? So now we turned into a system where we have an ex vivo organ culture. We take a salivary gland from Drosophila that we can culture ex vivo, which gives us the opportunity to do both genetics, but also use drug treatment, so we can then manipulate these with drugs. And when we don't add wingless, so this is now genetically inducing Wnt expression, you find kinesin and IFT140 co-localizing, but beta-catenin stays at the membrane where it should be, and it's degraded in the cytoplasm. If we now add wingless, genetically, we can turn it on. You have kinesin and IFT140 still co-localizing, but now you have beta-catenin co-localizing with them in the triple label shown here. So that's very revealing. And we see the same with tubulin, where we mark tubulin here in green, IFT140 in red, and beta-catenin in magenta. And again, without Wnt, magenta and beta-catenin is just at the membrane. In the signaling context, what's released from the instruction complex is localized together with these with tubulin on microtubules. So this argued very strongly that there's a complex that forms upon Wnt signaling on microtubules. And this is just one slide to sort of summarize a lot of work. So we know now that kinesin, CAP3, associated with microtubules, the IFTA complex. The business end here is IFT140 that binds both to CAP3 and to armadillo beta-catenin. And these are a combination of either pull-downs, immunoprecipitation from the in vivo tissue, or in vitro direct pull-downs where we can really show certain interactions being specific. So I'm not going to go in detail, but this is basically what we get from this as data. So then we ask the question, so what does it really do mechanistically, right? So in that assay, which is, again, it's a great assay because we can easily see nuclear beta-catenin when we add wingless. This is E-cadherin, and beta-catenin and armadillo normally co-localize at the membrane, but now because Wnt signaling has been activated, we see increased Wnt levels in the cytoplasm, and we see nuclear beta-catenin overlapping with a nuclear marker. And this is sort of enhanced by adding leptomycin, which inhibits nuclear export, so we can quite easily see nuclear localization of beta-catenin. And now we do the same experiment. So this is, again, the control here, up here. But we, at the same time, we knock down either IFT140 or kinesin, and there is no more beta-catenin in the nucleus. Everything else looks normal. So it strongly suggests that this IFTA kinesin complex is required for nuclear translocation of beta-catenin. And what's even more revealing here is that when we make, here everything is present, but kinesin has a mutation in the motor domain, so it can bind microtubules, but it cannot move. And so there is a bit more cytoplasmic beta-catenin detected because probably it gets kind of bound inside this context. But there's no nuclear beta-catenin enriched, again, as compared to the control, suggesting that nuclear translocation requires movement along microtubules. And I will show you some, how am I doing on time? Okay, that's good. I will show you some movies to actually really document that this is actually really happening. This was all Drosophila specific, so we did both the genetics and this initial biochemical and co-localization assays in Drosophila, but obviously we wanted to show that this is a conserved mechanism. So we asked the question, can we look at mouse cells or mice in general to find a similar mechanism? So this is now experiment done with mouse embryonic fibroblasts that are either wild type or mutant for IFT140 in the upper section here. And this is the control, and this is important control. This is IFT88, which is a key factor in the IFTB complex, right? So in both cases, IFTA and IFTB mutant, you don't make cilia, but the complexes are different. One is required for one set of transport, the other for the other. So what we see here, we add Wnt3A. This is the control without Wnt. We see nuclear localization of beta-catenin, and on the Western blot, you can see high levels of beta-catenin in the control. In the mutant, we don't see nuclear beta-catenin in the fluorescence, and we see very little nuclear beta-catenin in the loss of function IFT140. The control here is the same experiment, so this is wild type, this is mutant, and in both cases, for IFTB, in both cases we see nuclear beta-catenin, and we see high levels of nuclear beta-catenin also in the Western blot fractionation. So this shows us two things. First of all, the IFT140 IFTA requirement is conserved, and you do not need cilia because in this context there's absolutely no cilium because IFTB is completely gone. So no cilium needed for Wnt signaling, but you need IFTA complex to be there. And this might explain some of the confusion in the Wnt field. If you look at the literature in the mammalian Wnt field, you can look at 10 papers and you probably get 9 different indications or answers. Some say Wnt signaling requires cilia, some say Wnt signaling is inhibited by cilia, some say Wnt signaling doesn't care about cilia, etc. This clearly shows why that might be. Depending how you knock down or remove the cilium, you get different answers. Because it doesn't depend on the cilium. Here, there's no cilium, but signaling works. It depends on a specific set of proteins, which are the IFTA complex. So we could show nicely that this is conserved. And then we had one observation which was striking and sort of puzzling. And now we kind of think, we think we know why. There are different ways of making stable beta-catenin. One is to delete the N-terminal region by deleting the sites that are phosphorylated. And this is reflected by ARMS10, which is the original isoform that was generated by Eric Wieschaus and Mark Pfeiffer, where they deleted the phosphorylation sites. It's a 53 amino acid deletion. This is a more sophisticated mutation. It basically makes the threonine, that is the initial priming phosphorylation site, into an alanine, and it doesn't get phosphorylated. And in both cases, if you express those near the wing margin, you get many senseless positive cells, which you can see here in the staining. And so you get many sensory bristles, so it looks like a very sort of hairy wing instead of just having the one row on each side. And in the recruitment assay, we see nuclear localization of ARMS10 and nuclear localization of this ARM star, which is this mutant. But now when we remove kinesin or IFT40, the deletion ARMS10 still goes to the nucleus and it still activates the pathway, versus the star mutation is suppressed by these mutations, and also nuclear localization is strongly reduced. So this suggested there's something going on with this region of these 53 amino acids. And so Lynn looked at it in more detail, and so what you can see here is that actually those 53 amino acids are both necessary and sufficient for armadillo beta-catenin to interact with IFT140. She also confirmed that with the mammalian, with the mouse beta-catenin. And so the fact that the deletion still goes efficiently into the nucleus, even when there's no IFT140 or kinesin present, suggests that there might be a competitive binding to this region by an inhibitor that blocks nuclear translocation or sequesters beta-catenin in the cytoplasm. So we're looking for that, obviously. So the model from these studies suggests 2 things. That, first of all, this region is critical for binding to IFTA, for nuclear translocation, possibly by movement along microtubules, or likely by movement along microtubules. And there might be other factors, in this case we call it factor X, that also can bind to this region, and they would be cytoplasmic retention signals. And so in that context, that raises 3 questions, and this will be the last couple slides to sort of address that. So one question is obviously that this fragment, or this region, should act as a dominant negative in the pathway, because if you express it, it should bind both the positive and the negative regulator, and it should sort of soak up the system, and so the endogenous beta-catenin would not be able to bind and wouldn't be able to go into the nucleus. So that's point number 1. Point number 2, because we think there might be a factor X, there could be other proteins binding to the same fragment in the beta-catenin protein. And the third question is, so we think it moves along microtubules. Does it really? What's the mechanism? So I'll give you quick answers to all 3 of them. So yes, it does work as a dominant negative. And when we express low levels of this fragment along the wing margin, we start losing the wing margin, which is, again, consistent with that high level of Wnt signaling is lost. If we add more beta-catenin, we can suppress it. So consistent with the idea that it's the levels of beta-catenin. It's a competition between this fragment and wild-type beta-catenin. And again, genetically, this all makes sense. So it does behave like a dominant negative, and it is sensitive to the levels of endogenous or full-length beta-catenin. It does it so by blocking nuclear translocation. So again, this is with the control here, where we see nuclear armadillo beta-catenin. If we now co-express it with this short fragment, the 53 amino acids, we add wingless. So this is the control. This is with wingless. It doesn't go into the nucleus because it's blocked. And the same, again, we repeated this, or we reproduced this with mouse beta-catenin in the mammalian system using the mouse MEFs, and again, it's blocked as well. So it's a conserved mechanism from flies to mice, and we basically think that this fragment inhibits the pathway by blocking nuclear translocation. The second question was, are there other proteins to bind? So we use sort of the easy classical tap-tag analysis where you have two-way purification. And this just shows one gel. Lynn has done this several times, many times actually. So this is the fragment. This is full-length beta-catenin. This is the original protein extract, first run through eluate, etc. And so when we just look at this short beta-catenin fragment, this is now in human cells because the biochemistry is easier done in human cells. So these are HEC293 cells. So it's all human proteins. Beta-cat is here. We have IFD-140 we have here. This is documented. But then we have 4 additional bands, and we don't know yet what they are. Because of the COVID pandemic, using the proteomic facility at our place was complicated, so we don't know the data yet. Hopefully in the next month or the next 2 months, we'll have indication of what these are. And hopefully that will tell us if some of them might be a cytoplasmic retention signal or give us other insight into this mechanism. So that's exciting as well. And the third is now, what's the mechanism? Does it really move along microtubules? So this is now a movie where we have beta-catenin labeled in red, kinesin in green. This is one of these salivary gland cells. And if you look at the movie, you can see, for example, here, an overlap between armadillo beta-catenin that moves towards the nucleus, then sort of gets hand over, and then it kind of disappears into the nucleus. The red here marks the nucleus because beta-catenin accumulates. And if you look at this still here, you can see this dot, which is co-labeling beta-catenin and kinesin, moves toward the nucleus, hits the nuclear membrane or pore, kind of dissolves, goes inside, and then it's gone. So suggesting that microtubules really transport beta-catenin. There's one caveat to all of this. If you are a microtubule expert, you probably think already that you should ask me this question. Kinesin is a plus-end-oriented motor, and plus ends in the textbook are generally drawn near the cell periphery and the minus end near the membrane, near the nucleus. And so you go like, okay, that makes no sense. The plus end, the beta-catenin should be moved towards the membrane, not towards the nucleus. If you talk to like real microtubule experts, they tell you that's a little oversimplified and that you find microtubules of both polarities, plus ends, at the membrane or at the nucleus, and so it probably does sense. So what Lynn then did, she stained these cells for EB1, which is the plus end marker. So EB1 GFP shows your the plus end, and if you actually now do this experiment, this is without wingless, again beta-catenin just at the membrane. Here, some microtubules plus end labeled. We add wingless, we see beta-catenin in the nucleus, but we also see, if you overlay it, we also see plus ends inside the nucleus, which is the triple label here, which is white. And we also see tubulin, so microtubules going into the nucleus. These are confocal sections, so we're really looking through the nucleus here. Here's a plus end and tubulin, so part of the red stripes, which the lines which highlight microtubules are also green. And some of those green spots are in the overlap again with white is inside the nucleus. So this data indicated that plus ends of microtubules go into the nucleus or found in the nucleus in the presence of wingless, but not in the absence of wingless. We don't see any EB1 positive cell inside the nucleus. We see them near the nucleus, but not inside. And this is a movie showing sort of the EB1 in the control, no wingless, and this is with wingless, and this is the nucleus. These big black dots are the vacuoles that are the secretory vacuoles. This is a salivary gland, so there's lots of secretion. But the nucleus here is the bigger dark spot. And you can see a lot of plus ends near the nucleus. So there are definitely plus ends near nuclei. But what was totally puzzling is that when you add wingless, you can see EB1s going into the nucleus. And to make it more convincing, this is an experiment where we label the nucleus with H2, with histone 2 RFP. This is now in HEC293 cells, so these are mammalian cells. Win3A minus, you see plus ends around the nucleus, and you can see them move around, but they don't go inside. We add Win3A, and you can see, it's a little overexposed, but you can definitely see stuff going into the nucleus. So plus ends somehow, and this is really the big mystery, somehow they get through the nuclear pore, presumably, and microtubules penetrate into the nucleus. And the latest experiment, which I don't have a movie of, that Lynn made a movie, she can see beta-catenin with the plus ends moving into the nucleus. So it seems that there is a quite interesting mechanism now used for beta-catenin nuclear import that might be also used by other proteins. This is just sort of probably the tip of the iceberg, and we have to look at that much more closer. So I will close here with just summarizing this. So in this context, we can say that definitely this fragment is critical, and it acts as a dominant negative. It most likely binds, I mean, it looks like it binds other proteins. We don't know yet what they are, but some of them might be antagonizing IFD-140. And the mechanism really seems to be transport along microtubules into the nucleus. Of course, we need to look at this more detail. We need to do sort of 3D reconstruction from the microscopy with light sheet microscopy and other contexts to get a better 3D view, and also maybe label, or not maybe, to label the microtubules and nuclear pores to see if there is sort of a, if the nuclear pores open up and microtubules go in, etc. So this is still very puzzling because it's not what you find in the textbooks, and we'll have to look at it in more detail to really understand it. So I'll give credit again to the people who did the work. So this is a pre-pandemic picture, and this is the most recent picture where we actually had a picnic in the park when it was allowed in May, with low infection rates back then. Some of the work that I talked about was initiated by genetic screens from Ursula Weber, who did beautiful genetic screens in the context of Diego and Prickle, and she's now gearing up to look at disheveled in a similar context. Chema Carvajal showed very nicely that the centrioles are a key localized component downstream of PCP. Ashley did all the work on the SAR kinase and the phosphorylation, the differential binding between disheveled and Prickle on Wango. And Lynn joined the lab after, oh, I forgot, well, so Sylvie Balmer identified IFTA, and then Lynn basically took it. The last few slides were all Lynn's work, both biochemistry and establishing these assays for the movies to kind of start showing what's going on with beta-catenin and microtubules. And let's see, oops, sorry. This is where we work, so we are in this dark building behind, and we have beautiful views from the building. This is a sunset from our admin office, because we basically look down on Midtown Manhattan, and I'll stop here and take questions. Thank you very much.
If it's functional. Mm-hmm. Okay, so the talk is open for questions.
Please. Make him run.
Thank you for the interesting talk. So my question is whether wingless and Wangle competes for the same interaction site on frizzled.
Can you repeat the question? Maybe you have to take off the mask. It's a little muted.
Yeah, so wingless and Wangle, do they compete for the same interaction site on frizzled?
Good question. In principle, well, I have to say most likely yes. The CRD domain, we haven't mapped it further because the CRD domain is kind of complex and it's very difficult to chop it into pieces. But the CRD domain is the region which binds to winds, but it's also the region or the domain that binds to the extracellular loop of Wango. So based on that, yes. What we think is going on that in canonical signaling, wind complexes LRP56 and frizzled, with frizzled then intracellularly and LRP56 interacting with axin and disheveled, and this gets internalized to form signalosomes. So that is kind of counterintuitive. So if wind binds to frizzled, it should go inside, right? But in this case, it stabilizes frizzled on that side of the cell, because frizzled in the PCP context is physically associated with flamingo, with the cadherin, and that makes it stay at the membrane. So you basically enrich for frizzled to the side where the wind is coming from. And so it'll initially bind to wind to get sort of stabilized there. And then through this sort of amplification of the polarity, it then probably exchanges the binding for a Wango binding. This is a little hand waving because it's hard to do this, but I think we're starting to get some tools to kind of be able to look at it over time and have a better resolution.
But does that mean that's why to keep the wind expression low at the nucleus of the Pcp signaling, and then once you move outwards, the wind expression increases means canonical signaling is more active? Is it?
Yeah. I mean, in Drosophila, it's really complicated in a sense because the wingless gene, which is the main wind we have, is doing both, PCP and canonical. In a vertebrate or a mammalian context, you have multiple winds, and some are more dedicated, some are fully dedicated to canonical. They don't do PCP at all, based on existing data. And others are much more dedicated to PCP. So you can imagine there that depending which wind binds, you get different responses. In Drosophila, we have to deal with the problem that the same wind will do both things. It depends to an extent who will complex with whom. So if you have LRP56 or flamingo co-localized or co-bound with frizzled, you trigger different responses. So it's a little tricky to really, Wind4 is great for us because Wind4 has very little capacity for canonical signaling. So when all these gain of function assays and all these sort of manipulations we can do with Wind4 without having the problem of dealing of overactive beta-catenin signaling. So we might be able to tweak that a little bit.
Yeah, so I'll ask one more. From Royal Nuse's work, so he shows that these stem cells are the ones that they start producing wind ligands, and then the signaling increases from the nucleus. So he shows that it's because of the stem cells itself. So what about the PCP signaling? I mean, what is so special about those cells that they initiate this pathway?
So there's no real, so basically the wind expressing cells, they are there to organize the tissue, right? So they usually use not only to do PCP, but mainly to activate sequentially or by level of signaling different target genes of the canonical pathway. So it's basically using the same expression system. And I sort of simplified it. Of course, wind is at the margin, but wind is also present at the hinge where the thing is attached. And there's actually sort of competition for the wind from the hinge to the margin. And in other tissues like the eye, you have two sources of wind, and the cells orient towards both sources. There is also expression of a second PCP system, fat axis, which helps to coordinate the direction of this thing. So in many contexts, you might have more than one wind source, but because of the presence or absence of some of these other factors, the orientation will be still coordinated. So it doesn't really matter. So you could think that in the context of a stem cell where you have the wind source affecting the stem cell, it could also orient the mitotic spindle, or it could affect the asymmetric differentiation of a stem cell giving rise to a progenitor and keeping one of the daughter cells, keeping the stem cell identity. That's all compatible with our data. We just haven't looked at it in a stem cell niche context. But they are actually in the hematopoietic stem cell context, in the bone marrow niche. There's been nice work from a Japanese group, I'm blanking on their names now, apologies, that have shown that flamingo seltzer is critical in orienting the division spindle and basically maintaining stemness for one cell and then giving progeny. So there is evidence for such a PCP-dependent, wind-dependent orientation context. Yeah.
Basically, if I understand it correctly, like that the fact that there is only one wind in Drosophila, it means that whether it's like this canonical pathway or non-canonical pathway, it's regulated by the amount of wind, or like how close the cells are to this margin?
So I think it's the levels and the timing. So for PCP, you have wingless and Wind4, which is a bit more wind than the canonical is totally happy with wingless. If you remove Wind4, not much will go wrong, or nothing will go wrong. There is enough wingless. PCP, you need both, but they're redundant. So it could be that the levels are kind of, you have more wind that can bind to frizzled. And if you look at the affinity of the receptors, wingless has a 10-fold higher affinity for frizzled 2, which is the bona fide canonical receptor. Wind4 and wingless have the same affinity for frizzled. And while there is some redundancy between Frizzled-2 and Frizzled for canonical, the fact that the tenfold higher affinity would suggest that winglets will do all the business with Frizzled-2 and kind of leave Frizzled alone, unless there is no Frizzled-2, and then Frizzled-2 can step in. So the selection, which receptor is used or which pathway is activated, might come at the level of the receptor, naively, just by these affinity differences. And then there's the timing. So the canonical pathway sort of goes first, and PCP follows, because you first define the cell fates, and then you orient the cells.
Okay.
Does that answer your question?
Yeah, thanks.
Okay.
And another question?
Sure.
If I can have
You asked a lot of questions already at lunch. He's a smart
sorry.
kid. He was hammering me all lunch. I had barely time to eat.
So I was wondering, is there like a role for Beta-Catenin also in Pcp signaling? Because Pcp orients it's localized mostly to these adherence junctions where there's lots of cadherins. Pcp proteins are known to regulate cadherins, and cadherins are stabilized by binding to actin cytoskeleton through Beta-Catenin, which basically forms, as we could see in the pictures, it forms like two pools in the adhesions, and then if we activate in signaling. So is this...
Good question again. So, no, absolutely right. So there's actually a whole story in the lab that I didn't talk about because it would not be possible to fit into one talk, and it would require its own introduction. PCP signaling regulates through some of these kinases and other factors the stiffness of the tissue and cell adhesive dynamics. So we actually have a paper that just got accepted last week in Nature Communications that shows that PCP regulates tissue fluidity at the level through E-cadherin and associated proteins, and Beta-Catenin obviously links E-cadherin to the actin cytoskeleton, so there you have it, right? So there is some effect for sure in that context on PCP effects through Beta-Catenin. But that's really through the cadherin effect, really. So it's sort of indirect, but yeah, these proteins are kind of doing a lot, right? So. Yeah, sure, maybe. Certainly, I mean, it's coming in. Yeah, go ahead. He was first.
Okay, thanks for a beautiful talk. I was just like a bit of follow-up on this. So to how big extent you would say that the PCP pathway is like self-propagatory, and to how big extent it does need the gradient like all the time?
Good question. It's been actually proposed that you don't need the gradient at all, that you would just kind of trigger asymmetry through localized expression, and then it kind of gets propagated. If you look at the timing, when we see polarity first and how it propagates, it actually comes up first in the area of the wing where the gradient, the wingless gradient, as we can detect it with extracellular antibody staining, is the best. So it's in the middle. And then it kind of starts spreading towards the margin and away from it. So that would argue for a gradient being sort of important, at least for a certain time window. And then it propagates by just the regular sort of cell-cell interaction. If you make these manipulations where we misorient cells by genetics, right, either putting too little or too much of something, so Wnt4 overexpression or frizzled overexpression or frizzled removal, this propagates easily over 5 to 10 cells. And there's no gradient there. It's just a group of cells makes a wrong decision and it influences the cells around. So you can definitely polarize a field of 5 to 10 cells without a gradient. But the initial polarity is sort of in the area where the gradient is the most obvious. Let's put it this way.
Yep, like one reason to ask for that is also that there is a mechanism how to polarize the tissue over a bigger area, which is the lateral induction by notch signaling, right? Which goes actually from the other direction of the margin.
Yeah.
So there was like basically why the system is used, in the nature, like an alternative system to something that is already existing and can provide this directional cue. So it was like
yeah, no, I agree. I mean, if you look at other tissues, it's kind of modifications of the same. It's never the same. It's always some variations. If you look at the abdomen or the leg, etc., in the leg you need involvement of notch signaling to some extent because for the propagation you need it. In the eye, obviously, frizzled acts through notch as well. indirectly, etc. So you kind of take advantage of what the tissue gives you as a signaling pathway, and then you kind of use other tools that are available. So it's, yeah, absolutely. I mean, it's not, of course, in the talk I kind of have to oversimplify, otherwise we'll be stuck at the first part of the talk and never make it past that. But yes, there is different ways of doing this.
Thanks a lot.
Now you're going to have a cilia talk. Cilia question?
Yeah, I actually have a bit cilia question. Not silly, though, hopefully. So I have a question around the data on IFTA complex. So if you think, let's say, if you think there is some kind of competition between the cilia and the beta-catenin for the IFTA, or it can actually do both with the same, I mean, both at the same time, both these processes, so participate in ciliogenesis and regulates the Wnt signaling, or there is some kind of, I mean, you have to choose if you want to regulate cilia or you want to regulate the beta-cat.
Good question. So I don't know if you have to choose. So in the experiments where we don't lose the cilium, right, in the positive control, where we use Wnt3a to induce, the cilium is there because IFTA is there, IFTB is there, and it still works. So it's not a competition. So you would think, okay, if you lose the cilium, you push towards Wnt signaling. If you have Wnt signaling, if you have the cilium, Wnt signaling would be repressed. So maybe in some cell types that could be. It's possible. That's why also there's confusion. The relationship between Wnt signaling and cilia is complicated, as you know very well. But I think it's not necessary that it's a competition. It might depend on how much IFTA there is, or you can. What's clear from our data that the IFTA that we need for Wnt signaling is non-ciliary, and you don't need cilia to activate the Wnt targets. I mean, I didn't show that for reasons of time, but axin 2 is the endogenous target of the Wnt pathway, and in all these assays we can use it. In addition to looking at nuclear beta-catenin, we can look at axin 2 induction, and we see it as we would expect or not. So the absence of a cilium, let's say through IFT88, when IFTA is still present, doesn't look different from the wild type. True, that's correct, yeah. That could be. We haven't looked outside of sort of simple cells. But yeah, no, absolutely. Yeah.
I have a question concerning the IFT 140 interaction with beta-catenin. Is it somehow affected by beta-catenin phosphorylation? Because it looks from the data that beta-catenin has to be unphosphorylated for the interaction. So is it true? Have you checked it?
So good question. So we don't have a phosphorylated beta-catenin in that area because it gets degraded. So we could, I don't know, we have to see, engineer a phosphorylated beta-catenin that's kind of protected from degradation and then see if it still binds to IFT 140. Yeah. That hasn't been done. So we can maybe block the proteasome, or I don't know. I mean, it's possible. You can sequester it in a way that you can keep it away from the degradation. It's definitely the area that is phosphorylated. So the question is very, it immediately jumps to mind, but we haven't really done the experiment yet. But it's something that should be done, absolutely.
Yeah, thanks.
Thanks.
Just a follow-up to this question. The experiment with the fragment, would you say the fragment is phosphorylated?
If the short fragment is phosphorylated, we don't think it's phosphorylated. We don't think it's phosphorylated because it doesn't get recruited to the beta-catenin destruction complex. That we can say by co-localization assays.
Okay, I don't see any more questions. So we'd like to thank you again and thank everyone for the discussion.
Thank you very much.
And if you still have more questions, there is still a chance to meet Marek tomorrow in case you're interested.